Hello, my name is Vitor Piro from the Hasso-Plattner Institute in Potsdam, Germany, and the title of my work is ganon, precise metagenomics classification against large and up-to-date sets of reference sequences. Let's start talking about data growth. Here in this plot we can see the date of growth on the last 10 years for three major NCBI repositories. And important to notice that the growth is continuous and uninterrupted. However, when we are thinking about metagenomic studies, raw data is not so important as diversity, so we need to know how much diversity those big repositories have. And we can see that in this table where I list the number of species represented on RefSec for also the last 10 years. And we can see, if we calculate, there is a yearly growth rate of 23% a year, which is a very big number. If you take two data points from 2018 to 2020 in an interval of 2 years, there were more than 23,000 species deposited on RefSec. If we go from the beginning of this year until the latest RefSec release and calculate it, we could say that there were 18 new species added to this repository on average a day, which is a very big number. Our motivation was to provide a methodology that can make use of whole of this data. As well, to keep up with the data growth, as I just showed, the number of species has been growing constantly over the last years, and we need a method that it's able to keep with this growth. Another idea was to avoid pre-built or default databases, which are common practice in metagenomic studies, but they give the false sensation of being using recent references, where they in fact are usually not updated by months or years. Final point is that we wanted to also have a methodology able to explore and combine references dynamically, not having a big static index where we have to use for doing our analysis. For those points, we came up with a method called ganon, which makes precise metagenomic assignments against large sets of reference sequences. It can be found on GitHub, and ganon provides an efficient indexing methodology for large sets of references. It can keep up with the data growth by first doing those indices very quickly and also make them updatable. We avoid the use of default databases by having an ISCB script for creating custom databases, and also a way to flexible use them in a combined way, making the tool very, very strong for data exploration. And we can go from raw reference reads and reads to results very quickly. So how does ganon do that? Let's go to the steps. First, having some set of reference sequences, we're going to have some taxonomic information, and this taxonomic information is going to be used to cluster those sequences together. Those clusters of sequences are going to be decomposed into k-mers, and those k-mers are going to be inserted into our main data structure, which is called the interleaved Bloom filter. Once we have this, we're going to decompose a set of reads that we wanted to analyze in the k-mers the same way the references were decomposed, and with our classification method we're going to try to tell the orange or if one of those reads based on our reference sequences. Now let's go into detail in each one of those steps to see how ganon works. First, clustering with TaxSBP. TaxSBP can be seen as a metadata clustering. We don't use any sequence to make those clusters. What we use is actually just the taxonomic relations or taxonomic proximity between sequences. TaxSBP does that by an approximation algorithm to trying to solve the hierarchically structured bin packing problem using the taxonomy. And what we want to achieve here is to have clusters of equal size and with a fixed taxonomic rank, which are close together on the taxonomic tree. So for example, if you look at this image, we have a small example of part of a taxonomic tree where we want to make clusters at the species level, which are the circles colored. We have here 5 species represented. And what we want to do is to put them in clusters of equal size. So the squares representing the sequences are going to be separated into groups and they have to have equal size. What TaxSBP is actually doing is trying to put those sequences in those clusters in a way that those sequences that are closer together, united by a common ancestor, are going to be clustered together. For example, here in the example of sequence 4 and 5, since they have a common ancestor, they would be put together in the same clusters. And that's a small example what TaxSBP is doing by using the hierarchical structure of the taxonomic tree to make an informed clustering. Once we have this, we're gonna insert the elements of our clusters into our main data structure, which is comprised of a Bloom filter. Bloom filter is a probabilistic data structure to evaluate set membership. It comprises of a bit vector and a set of hash functions, and that's how a representation of a Bloom filter looks like. A Bloom filter is basically a bit vector of zeros, which is populated by setting ones on positions on the hash function. So first we're gonna get our reference sequences, decompose them into k-mers, and populate this Bloom filter with ones for the matching hash functions. Once we want to query this Bloom filter, we're gonna do the same for our reads. We're gonna decompose them in k-mers, but we're not gonna insert elements anymore. We're just gonna check for the same hash functions if they are able or disable, and we can tell if an element is present or not. However, on this case presented before, we don't have just one big Bloom filter where we want to see if our sequences are matching. We have several groups of filters, which would be our species in the example on the last slide. So we would need, in this case, several Bloom filters, one represent each one of the species we have. However, due to the very large amount of species or sequences available today, it would be impractical to do this approach. By querying several thousands of Bloom filters would take too long. That's where the interleaved Bloom filter comes in. Basically what the interleaved Bloom filter is, it's the combination of several equal-sized Bloom filters into one, interleaved bit by bit, as shown here in this picture. And without entering too much details of the technicalities of the Bloom filter on the IBF, it will enable us to query multiple filters at the same time and very quickly. In your case, each one of those the filters will represent a species group. Now the classification method. For each one of our read, we're gonna query the IBF, and we're gonna have a list of k-mers for each one of our subfeatures or species. With those, we're gonna apply the k-mer counting lemma to get the number of shared k-mers which are necessary to confirm occurrence of a certain read in a certain subfilter, considering a certain amount of errors. Once we have this set of matches, we're going to apply filtering for each one of those reads. So multiple matches for one read are going to filter first by selecting matches without errors, and if there's no errors, we're going to select matches with one error, and so on and so forth. And even though after those filterings apply, it may occur that we still have multiple matches with the same error rate. So we can optionally apply a lowest common ancestor algorithm to report those multiple matches into a single taxonomic unit. We also have a specific case for unique matches where we want to avoid low quality unique classification. So we're going to set up a more strict error rate for those. Last but not least, ganon can perform hierarchical classification among databases or reference sets. And that's possible in several levels and also in several filters. I give you an example. For example, one could build 3 different ganon indices for bacteria, viruses, and archaea, as an example and use them at the same time as if they were one filter. One could also create a filter for the human reference genome to remove, for example, host of your analysis, and then the remaining reads would be classified against the full RefSeq. One could also combine all of those together using ganon in a very dynamic way, where each one of those steps could be adjustable by error rates and can be run with just one command line. This approach is also very interesting and fits together with the updatability of the indices that ganon is able to. So one would create small indices for specific organism groups that can be easily updated. Just a method recap on everything we saw so far. First, we start with the taxonomy, and if you want to cluster, then in this example by the species group, we use TaxSBP to create equal-sized clusters which are rank exclusive. Next, we would index those clusters into several Bloom filters, which one of each cluster, and interleave them for fast queries in the interleaved Bloom filter. At the end, we would carry this IBF and with the k-mer counting lemma select possible matches and using filtering procedures to better cut the false positives from the true positives. Now to the evaluation methods of ganon. First, I want to present our own evaluation, which shows the influence of reference selection on the results. And second, I want to show an independent evaluation that was provided by the ISCB lab, where they have several metrics and several data sets to evaluate a larger amount of tools. First, let's start with our evaluation here. Let's start with the indexing. We had three main reference sets that we evaluated. We started with the RefSeq old, which is a reference set from 2015. It's very outdated because we want to evaluate how outdated data sets perform nowadays. And we also selected a recent set of the RefSeq, being the RefSeq all, the complete RefSeq for bacteria, archaea, viruses, and fungi organisms. And the RefSeq-CG is the same set, but containing only the complete genomes. For the RefSeq all and RefSeq-CG, we didn't use the complete set of sequences. We just kept the same diversity by keeping the top 3 assemblies for each one of the taxonomic entries. So we could reduce a lot the number of sequences in these repositories, but at the same time keep the diversity. And here is a table showing the indexing times and memory usage and index size for 6 tools compared, ganon included. And we can see on the RefSeq code, there's a database for 2015, the tools performed quite well, not exceeding more than 4 hours to index the set. But that was good in 2015. Data from today, meaning RefSeq-CG and RefSeq all, take way longer and use way more memory. And as you can notice here, ganon and diamond have very good indexing times, but on the RefSeq all, not all the tools are listed here. That's because not all of them could cope with such big amounts of data. And in this plot I'm showing exactly here, where on the x-axis I have the amount of base pairs in each one of our reference sets. And on the y-axis, you have the time that they took to index them in days. And you can see that Diamond, ganon, and Kraken 2 were able to scale with those data by indexing them under a day, where all the other tools really exploded in runtime. And why is that so important? Because when you look at the classification results, and you can focus here on the leftmost plot, where I'm showing the sensitivity values for one high complexity data set evaluation, is that the selection of your reference sequences, it's more important than the selection of tool itself. Not only at the time, for example in the RefSeq old, in comparison to the recent RefSeq, but also in the subset of RefSeq when you compare the RefSeq all with the RefSeq-CG. You can see that the difference in sensitivity is very strong, meaning that the choice of a reference set, it It's more important than the choice of the tool itself. But you have to make the choice of the tool based on the ones which are able to actually index such large amounts of data. Now, to go to evaluation of each one of those tools using the same data set, we're going to show a bit of the LEMMI benchmarking, which evaluated several tools in several different metrics, not only precision and recall, but also some others, including memory and runtime. And those metrics were calculated for 8 different data sets with different complexities and k-mer diversity. And at the end, each one of those tools is going to achieve a score, meaning that there's a harmonic mean on all of those metrics used. And those scores can be weighted by certain scenarios like pathogen detection or species identification. And those weights can be also tricked by a user online. So you can have your problem and tell what characteristics are more important for you, more precision, less runtime. And LEMMI is going to try to score those tools accordingly and show you the best one for your problem. By picking up 4 default scenarios in LEMMI website, we can see that ganon systematically is performing very well, being among the top 3 tools out of 8 to 10 tools compared. And that's very important to show the strength of ganon, not only by using very large amounts of data, but also when using the same data set that it's performing very well and competitive with the state-of-the-art tools that were developed so far. Well, that's about it. There's a message to take home. Ganon is a taxonomic classification tool for short reads designed for metagenomics. It is scalable and have a fast indexing procedure, and is the only tool among all of those evaluated that provides an update method to update those indexes in a fraction of time used to build them. I focus here on ganon as a tool for flexible data exploration, where it has this hierarchical classification capability, but also integrates very well with Genome Updater, which is another tool developed by us that can keep subsets from RefSeq and JBank updated. And that's very nice to have your own repositories. In the future, it's important to say that ganon is under active development. It's not a tool that stopped on its publication, and several next features are under development and are being released periodically. Like, we are trying to reduce a bit of our memory usage or our indices, also working on strategies for classification speed. We are planning a complete integration with the new taxonomic database. We plan to have an abundance estimation model and so on and so forth. Please follow us on GitHub. I'm very open to answer questions there and also accept feature requests. With that, I would like to thank all the people and institutions involved in this project, and I would be very happy to answer any questions now if there's some time. Thank you.